One. We're moving now from biochemical pathways to more of the molecular part of the class, the cell and molecular class, and talking about how DNA is put together. And one of the first things you learn about nucleic acids is A pairs with T and G pairs with C in DNA, A with U in RNA. And some of that came from experiments done by Shargoff, where he extracted nucleic acid from many different tissues, from many different sorts of organisms, and got a very consistent ratio of 1:1 for A to T, regardless of the type of tissue, and 1:1 for G to C, regardless of the type of tissue. Although the percent GC could be very different depending on the organism, so that's different numbers over here. Then Rosalind Franklin was doing experiments on X-ray diffraction studies on nucleic acid. This is one of her images. And she was in the same place as Watson and Crick. Watson and Crick went to talk to her, looked at her data, unfortunately didn't give her sufficient credit for her experimental data, and for that reason her name was left off the Nobel Prize. She's gotten some recognition now later in the day, but it's unfortunate she didn't get to see it while she was alive. James Watson is still alive. He's in his 90s. Of the Watson and Crick model, double helix model. He is unfortunately a racist and has repeatedly stated that there are genetic differences among races that are responsible for differences in intelligence among races, which is not supported by science and also despicable. And he has lost a lot of his many awards because of this, and people and societies and even his institutions have disavowed themselves of him. So their big contribution was the double helix model. And one of Rosalind Franklin's big contributions to this model was that the backbone that you're seeing here in the double helix had to be on the outside. They had it the other way around in the initial models that they were showing her. So really she is well deserving of the Nobel Prize. But there are no posthumous Nobel Prizes, and unfortunately she passed away. But in this model, you can see the sugar backbone here, and you can also see the origin of the orientation of nucleic acid. So you have a 5' end and a 3' end that is named for the number of the carbon in the sugar. So this end is the 5' carbon in this ribose, or deoxyribose, depending on if it's RNA or DNA, and the 3' sugar on this. Let's see, this is yeah, okay, so this is DNA. So new nucleotides can be added onto the 3' end. They cannot be added onto the 5' end. And so if this nucleic acid would grow, it would grow from this end and from this end here. And then the base part sticks into the middle, and this is where hydrogen bonding between bases happens. 2 hydrogen bonds between A's and T's, 3 between G's and C's. When you do a space-filling model of DNA, you get what's called major groove and minor groove. Proteins that bind to DNA and modify DNA's ability to be read fit into that three-dimensional shape of the major groove or the minor groove. There are other forms of DNA, such as Z-DNA, a different way of folding up, which is relatively rare, but it exists. We won't talk much more about it, other than you should know that it exists. Here's an H5P question for you. And chromosomes are made up of DNA plus the protein that wraps that DNA. A human chromosome is between 2 and 10 microns long. If you stretch out all the DNA in that, it would be about 5 centimeters long, or 5,000 times longer. And if you stretch out all the DNA that's in a nucleus, if you put all the chromosomes end to end and added that up in a human nucleus, that would be more like 6 feet or 2 meters, sorry. And obviously folded into very small compact shape that can be moved around. Chromosomes are not always in this shape. Chromosomes can be circular and then twisted up. This is a circular chromosome in a prokaryote that has been released from the prokaryote, and you can see its expanded shape there. And then it's within an area of a prokaryote called the nucleoid, but it is not associated with a great amount of protein as it is in eukaryotes, and it's not membrane bound. So if we think of prokaryotic DNA as circular, that circle can coil upon itself, and you get these sorts of shapes in electron micrographs of the chromosome in prokaryotes. This coiling pattern can get to be a problem in that it can, if you've ever had a bunch of rope, or in the old days when telephones had cords, they would fall into these super coiled sorts of patterns where it's hard to unknot them. Enzymes have evolved to do that, called topoisomerases. There's topoisomerase 1 that makes single-stranded breaks in the DNA and then repairs those to relieve stress on the DNA coil. So topoisomerase 1 breaks only one of the two strands, allows unwinding of that coiling, and then reforms the break, and so that it's intact. That's topoisomerase 1. Topoisomerase 2 for double, makes a double-stranded break. So it breaks both strands of the DNA, allows it to reform to release supercoiling in the DNA. So in this example, this loop is behind. You have action of topoisomerase 2 breaking both strands, and now it's repaired so the loop is in front. So again, topoisomerase 1, single-stranded breaks, topoisomerase 2, double-stranded breaks and repairs. And then the DNA is not naked DNA. DNA is wrapped on proteins. The protein plus the DNA is called a nucleosome. This is often called beads on a string, because it looks just like beads on a string. The string is the DNA, and the beads are the histone proteins that the DNA is wrapped around. So the histones are arranged like so. And I might ask you to draw this arrangement, extended response, on a test. And so let's make sure that we slow down and understand this. So there's different isoforms, different forms of the histone protein sequence that is related to each other, such that we know this is a duplication of a gene, but they're not exactly the same sequence of these different types of proteins. So you have H3 histone stacked on top of each other, H4 histone stacked on top of each other, H2B stacked on top of each other, and H2A stacked on top of each other, one there and one there, hiding in the back there. And then H1 histone is off to the side, spanning two histone globular subunits here. So the DNA wraps around this nucleosome bead 2 times, and then there is linker DNA between one nucleosome and the next nucleosome, and then you're on to the next nucleosome. So 8 histone molecules, 1, 2, 3, 4, 5, 6, 7, 8, actually 9 with H1 there. So 8 that are similar shape, and then a 9th one there. And about 146 base pairs of DNA to go around, and then plus the linker DNA, and then another 146, and so on. If we go from zooming out, so we have our DNA double helix that is now going to be wrapped around histone particles. This is just showing the 8 and not the H1 off the side here, the 9th one there. Okay, and then the linker, so on. Okay, this we can see on a micrograph, and that's about 10 nanometers across a nucleosome bead. Those will fold up on themselves, so each of these beads tightens up into another structure to form a different kind of strand, different kind of fiber. This is about 30 nanometers thick, so about 3 times thicker than just the width of one nucleosome. And then those fold up even further into these loops. You can see loops here in this micrograph of DNA. And these loops are about 300 nanometers thick. And then further, those loops are wrapped into heterochromatin, which is about 700 nanometers thick. And then finally, the heterochromatin is wrapped into what we associate with chromosomes, which are about 1400 nanometers across. This is a chromosome with 2 sister chromatids here. So putting it all together, the width of a DNA strand is about 2 nanometers, wrapped around a nucleosome about 10 nanometers, packed into a chromatin fiber about 30 nanometers, looped into loops about 300 nanometers, that folded into heterochromatin about 700 nanometers, and then a chromosome about 1400 nanometers. Okay, so we have 9 proteins associated with a nucleosome, and then the DNA that wraps around that nucleosome. Both the DNA and the proteins can be chemically modified. The histones are proteins, they can be methylated, have a methyl group added to them, or acetylated, adding an acetyl group added onto them. And this changes how the DNA wraps around the nucleosome, which changes how accessible that DNA is for transcription. So eventually you're going to want to use that DNA to transcribe a gene, and then that gene transcript will get translated into a protein, and then that protein gets used. In order to transcribe a gene, you have to have access to it, and if it's tightly wound, such as in that chromosome, you don't have access to it. Even at the level where some is unwound, if it's wrapped around a histone that has been methylated, that causes the DNA to be wrapped even tighter, meaning it is less likely to be accessible. And if it's acetylated, it causes the DNA that's wrapped around it to be looser, meaning that DNA is more accessible. These changes, okay, methylation and acetylation of your DNA can happen during your lifetime. These are things that can happen as a result of environmental exposure. Environmental exposure can be something like obesity, or exposure to pollution, or exposure to stress, physiological and mental stress. Mental stress that causes physiological stress. So things that happen during your life can change how accessible different parts of your DNA are, and changes the probability of whether or not you're going to be able to express a gene that happens to be wrapped around that histone. So things that happen in your life can affect your the likelihood that you're going to be obese, the likelihood you'll develop diabetes, the likelihood that you'll be tall. All those things can be affected by these modifications of the molecules that wrap up DNA. Normally we think of changes to the DNA itself, like changing out an A for a C, an actual DNA mutation, causing the sorts of things that will lead to some sort of physiological change. But it can also be changes to the molecules that wrap your DNA. Again, that can cause changes in transcription. These sorts of changes are called epigenetic. Let's see if I can write that. Okay. Pen. Epigenetic. I'm writing with my mouse pad here, so sorry it's taking so long. Epigenetic, it's all one word. And it means a change to your DNA, the accessibility of your DNA, that is not necessarily related to a change in the sequence of your DNA. It's not related to a change in the sequence of your DNA. Both things can happen at the same time. Epigenetic changes can happen across generations because there can be multiple generations that are exposed to the same environmental event that would cause an epigenetic change. An example, let's say a woman is in her third trimester of pregnancy and she has some sort of traumatic event. That's going to affect the possibility of modifying histones on her own DNA, on the fetus's DNA, and I don't know if this happens by the third trimester or not, but the developing eggs in that fetus. So that's 3 generations of humans affected by those epigenetic changes. Okay, I want to show you this video of folding. Let's see, here it is. Nope, that's not it. This is it. Okay.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is...
Okay, they're not showing you the H1 subunit, which is off to the side. Okay, later on they're going to not show you other proteins just because the animation gets too complicated. But the H1 number 9 is definitely there.
The nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body.
So again, it's not that each chromosome is 6 feet, it's that if you took all the linear chromosomes in a eukaryote and you put them one next to another, end to end, you would get 6 feet.
An object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope. Chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing.
Okay, so this is a little confusing. So the thing that you would see getting pulled to one side or another side during mitosis, that's a chromosome. And you can't always see that in a cell depending on what phase of the cell cycle you're looking at. But it's not to say that the DNA breaks up and only then condenses during when it's getting ready for mitosis. The DNA is always one, chromosome number 13 is always the same sequence, all connected from nucleotide 1 to nucleotide 5,292,000 or whatever it is. And that is always the case, regardless of what you can see under a microscope. It's only folded up into a shape that you could see under a microscope that we call a chromosome. Having said that, regardless of what phase of the cell cycle, geneticists always talk about the position of a gene on a chromosome, and they don't care at all what phase you're talking about. So this is a cell histology sort of term that they're saying they don't exist in all phases of the cell cycle. From a molecular biology point of view, they do.
At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.